I'm Rafael Lemaitre. I joined the museum in 1989, and I'm a curator of crustaceans in the Department of Invertebrate Zoology. I study decapod crustaceans, shown here on the left, which is to say shrimps, lobsters, and crabs, very diverse groups and very important groups with about 15,000 species. But primarily and particularly, I study hermit crabs. And most people think of hermit crabs as crabs that live in empty coiled shells, but in reality, they're very diverse in their use of housing. Some, like here, use rocks, bryozoan colonies, pieces of wood, straight shells, or even can burrow in corals. Others have made arrangements, so to speak, with anemones or live in clam shells or soft stones or even sponge matrix. Their morphology is quite odd. Once you remove them from their housing, they look like halfway crabs attached to some kind of worm-like part. They are imperfect in the sense that they're asymmetrical. They have appendages usually only on the left side, but with a lot of variations. This imperfection is what makes them so interesting, because they're actually adaptations, and it's like we've caught them in the act of evolution. Their coloration can be quite spectacular, such as these species of aniculus from the coral reefs of the Pacific. They can range in size from Burgos latro, or the coconut crab, that can be about 16 inches long, 3 feet in span, and weigh 8 pounds, to most hermit crabs, which are about an inch or 2 centimeters more or less long. Now, taxonomists like me are generally obsessed with finding out how many species really exist on this planet. This is a big question for us, and for many reasons. In case of hermit crabs, we know so far there's about 1,300 species or so, although we guesstimate that about 600 still need to be discovered. But where to look for these? Well, as I was pondering this question one day at the beach, I found a bottle full of shells, and when I dumped the contents, to my great surprise, a genie came out. And like in the legend, he told me I could ask him 2 questions. And of course, I didn't hesitate. And I asked him, well, how many species are there, Genie? And where can I find them? And he said, drop. And I said, excuse me? Oh, yes, of course, Genie. I said, are you telling me that I need to call Carl Baldwin and join her drop program in Curacao? And so I immediately did that. And the Genie was right, because in Curacao we found a spectacular fauna of hermit crabs with very colorful species, although most of them were actually fairly large in size and well known, although from very few faded museum specimens. So I was just very excited to see them in life for the first time. Then something interesting happened, like it usually does in science. My IZ colleague, Jerry Harrisewicz, who's a malacologist, started to notice, while he was down in the sub, that the sea bottom around Willemstad in Curacao was littered with numerous glass bottles. And many were filled with tiny shells, although the shells were nearly always empty. In other words, without the mollusk animal. And so he noticed that some shells had live hermit crabs. And he concluded that the octopuses might be dragging the shells inside the bottles to feast on the mollusk and the hermit crabs. Then he began to retrieve bottles on a regular basis to study the snail shells, and soon the lab started to look like a bottling factory. Amazingly, he discovered some new shells among the bottles. He then passed on the hermits on to me, those lucky uneaten hermit crabs, and I and my colleague Darrell Felder started studying them. A rush then ensued to obtain more bottles at every dive, and it was really tedious work to sort through hundreds of shells and extract them from their shells. I was really puzzled by their size, and I was evidently looking at adult specimens because some of the females had eggs, and moreover, a lot of them had some peculiar morphology and even different colors that I don't have time to go through, but they turned out to be undescribed species with no known relatives in the Caribbean or anywhere else for that matter, so that meant I had not only new species but new genera. Furthermore, we started finding these same kinds of hermits in the autonomous reef monitoring structures that Nancy Knowlton was putting down in Curacao. So it was quite evident at this point that the species found on the bottles and the arms actually live in very cryptic habitats of the reefs where biologists had seldom looked carefully for hermit crabs before. Then we found them in Guadeloupe, on the island of Guadeloupe. Actually, the Paris Museum team went out there to sample for micromollusks using suction devices and all sorts of neat things, and they found some more of these species. And so here's a comparison with a penny to give you an idea of the unusually small size of these hermit crabs. The size is really at the boundary of the microbenthos and even the meiobenthos. So in the end, I ended up with 6 new genera and 8 new species. And so why is this discovery important if it's not really unusual to find new species? Where it's unusual because we're finding them in very cryptic habitats and only 3 of the more than 7,000 islands of the Caribbean. And so it is probable that the pagura diversity is much more than we think has been highly underestimated. In other words, this is like discovering the insects in the canopy of the forest. So I really thought that, once the genie is out, we can't put him back in. And we really have a job to go through now to find these new species and et cetera. Thank you very much.
You said that some of the shells had been recently inhabited by hermit crabs. How can you determine that?
Well, because they tend to be worn out in many ways. So you can determine that that's the case. The hermits do tend to do things inside with their shells to make them comfy. So.
The genera and species you described, that's based primarily on morphology, exclusively on morphology. Maybe I missed that.
Yes, it's
repeat the question.
Strangely so these days. Yes, it was exclusively on morphology, although, and I've probably failed to mention it, but the beauty of the program, research program, is that we've collected all the genetic data as well. So we can now, analyze all this genetic information to, test morphological thing as well.
One last question, that's for Rafa, and I think everybody's dying to know this. These hermit crabs, they spend most of their time, in the shell. Why are they so colorful? Even the back portion of them.
Well, a lot has been written about the significance of color, as you know, for fishes and invertebrates and all sorts of things, and about the evolution and significance of color. And there's there's mimicry and there's deceptive patterns. They tend to want to look not like a hermit crab, but something different, so that avoid predators. But I think in the case of hermit crabs, although this hasn't been studied very well, if you think about it, these things live retracted, and if you want to find a partner and all you see is the hands of your partner, you better have a nice color that somebody can recognize. So I think there's been some studies about it. It has to do a lot with sexual selection too.